Talking about the pulmonary system. So we kind of moved into that from cardiovascular, and we're going to talk about ventilation and respiration. So pulmonary kind of refers to lungs. Ventilation is the act of breathing in and out, moving air in and out. And then respiration is generally referred to as the exchange of gases. So although sometimes these things can be kind of thrown around and used interchangeably, but that's technically what we're talking about. So the respiratory system. So the main function is to supply the body tissues with oxygen and dispose of carbon dioxide. So that's what we're doing with breathing. Now there are other things that can happen as a result of that, like pH changes, adjustments, things like that. But really, that's what the purpose of this is to do that gas exchange. So collectively made up of four processes: ventilation, which is breathing; external respiration. So external respiration is in the lungs. So you're exchanging gases with the environment. So we pull environmental air into our lungs, and then that exchange takes place in the alveoli, those little sacs in our lungs, and that's external respiration. Even though it's in the lungs, it's still with the external air, outside air. And then it transports those gases in the blood. So that's kind of a vascular thing, but you have to consider how the oxygen is being transported. For instance, it's transported mostly on hemoglobin. Meanwhile, carbon dioxide is primarily transported on the bicarbonate anion. So, and then internal respiration, this is the whole point of it, is to supply that oxygen to cells that are all over our body. So in our feet, in our liver, in our spleen, whatever. All of those cells that are functioning need oxygen, and they need to get rid of their carbon dioxide. Okay. So the anatomy of the lungs, we can see here that we have on the right, we have three lobes. So one lobe, and then this is a lobe, and this is a lobe down here. And on the left are only two. Okay, so the left as it's facing us, as always. But then we have this area, this little notch right here, which is where the heart goes. So that kind of takes up some space in the left lung. So if you look at an x-ray, you'll see that the left lung is smaller, and that doesn't mean that there's, some kind of what we would call like atelectasis or fluid or anything like that. It's because the heart is sitting there. All right. We also have the bronchial tree. So the bronchial tree, if it were upside down, I guess it would look like a tree if it were turned the other way. But you can see the windpipe, or the trachea here, has cartilage kind of supporting it, so it doesn't close. And then you can see that first branch here. You still have some of that cartilage, and then it kind of gets less and less. But it keeps branching, up to like 23 to 25 orders of branching before it gets to the respiratory center, or the respiratory, I guess, the alveoli and the respiratory bronchioles. All right, so let's see. We'll look at that, and we kind of have this graph here that kind of tells us some stuff about the branching. So we have the trachea. Trachea is that's your first order. But it's got some cilia. So the cilia are those they're like, I don't know if you've learned villi and microvilli, but the cilia kind of beat, and they will kind of move, and now on top of the those cilia, or around there, we have mucus. And so it's kind of these cilia are moving, and they're beating, and they're moving that the mucus with dust and dirt, whatever's been collected on it, organisms, pathogens. It's moving that out of the airway. Okay, so it starts at well, it doesn't start. It starts wherever the dirt is, but it goes all the way down, and we can see that we have cilia all the way down into the beginning of these bronchioles, and then it kind of goes away. And when you get down into these respiratory areas, then you don't have the cilia. Smooth muscle. So smooth muscle, remember, we have constriction and dilation. So we want to dilate to get more air in, and that's those beta 2 receptors. And when those are activated, and then we have parasympathetic. activation, which can cause constriction. Okay, so that's going to happen with these smooth muscles. When we talk about the smooth muscle, that's what we're talking about, and we'll see some pictures of that later. And we can see that that happens all the way down also to the bronchioles. But it's not. But the alveoli, where the functional respiration really takes place in the lung, they don't have anything. They're really just cells. They're kind of like, when we learned about let's see, venules, capillaries. When we learned about capillaries, we found that they were just like one cell level wide, just one little piece of epithelial cell. That's kind of how the alveoli are. So they don't have anything. So, and you can kind of see that. Nope, no cilia, no smooth muscle, no cartilage, nothing. They're just there for air exchange. They depend on everything else to kind of take care of them. So yeah, so terminal bronchioles branch into respiratory bronchioles, which divide into alveolar sacs. So we can see, or alveolar ducts. So we can see kind of some of these little bumps here that are capable of respiration, but really it's the alveoli that are doing most of it. So about 23 to 25 orders of branching, depends on what you read, and can be divided into the respiratory zone, and that's where gas exchange takes place. And that's just kind of something notable, because we see this and we see a lot of the bulk. So if we go back to this picture, we see a lot of this as, we see all these branches, and none of that is actually, there's no respiration, there's no gas exchange that's taking place in these branches until you get to those tiny respiratory bronchioles. But they are branching out, and most of the mass of the lung are actually these alveoli. So that's where the gas exchange is actually taking place. So that's 70%. And those things that we were just looking at, all of these branches of the bronchioles or the bronchi, that's only about 30%. So from the trachea down to the bronchioles, that's only about 30% of air is in those areas. The rest of it makes it down to the alveoli. All right, and trachea is also known as the windpipe. So microscopic anatomy of lobule of lungs. So what we're really getting to, so we have this picture over here. It shows the terminal bronchiole, shows some alveolar ducts and the alveoli that are kind of sitting in here. Okay, and there's a lot of gas exchange taking place. But it makes a lot more sense. I love this picture. So it makes a lot of sense. It kind of shows everything that's kind of going on where gas exchange is taking place. So we can see these terminal bronchiole that divide into the respiratory bronchiole. And we can see, and this is the respiratory bronchiole here, and we can kind of see that there are some of these little sacs that have formed that can have respiration, so they can have gas exchange in those areas. But then those bronchioles branch into and form these alveoli. And that's the primary side of gas exchange. So each lung has about 300 million of these alveoli, and each are very small, about 200 micrometers. And they also have pulmonary vascularization around them. So we have these these capillaries that go around the alveoli, and they just kind of cover it. And so as this blood is moving very, very slowly, so it's going to be going down in the blue, okay, because deoxygenated, and then it kind of moves past, and then it's exposed to the air as you're breathing, and then it gets oxygenated. And so it's moving past all of these to gain that oxygen and to drop off that carbon dioxide. Okay, and they surround, those pulmonary capillaries just surround all those alveoli. So pulmonary ventilation versus respiration. Okay, so we've already talked about it. Pulmonary ventilation, that's the act of breathing, inhalation, exhalation. Respiration, we also talked about this, is divided into external or internal. External is exchange of gases between the alveoli that we just looked at and the blood, and that's caused by passive diffusion of O2 and really CO2. Okay, so passive diffusion of gases. Okay. So the point is, it's not being pumped out, nobody's pushing it, it's just passively diffusing, going down a concentration gradient or pressure gradient. So internal respiration, exchange of gases between these systemic capillaries in the body and tissue cells, and it supplies cellular respiration. And of course, we need oxygen to make ATP. So that's really the main point of getting that oxygen down in there, is to run that mitochondria that is the powerhouse of the cell. All right, so measuring lung volume. So let's look at these lung volumes and capacities. So this is an old-time spirometer. They don't really look like this as much anymore, but it has this little, what they call a bell, that's kind of going up and down. So as you breathe air in, it kind of pushes that what they call a bell up. So this can go up and it can go down. And as it does that, there's this little writing device here that kind of just scribbles as this drum turns. Okay, so this drum here is turning around, and it just kind of writes what you're breathing here. So that's kind of interesting technology from the past. Now this is done a lot more in a more higher technology type of way, but that works. But what it does is it says as this person is breathing, they're breathing in and then they're breathing out, and that bell is going up and down and the drum is turning, and it gives us some information. So we call that just in and out breathing. We call that the tidal volume. That's about 500 milliliters. And then we know this, you could do this experiment on your own right now, and just after you breathe, after you finish taking a breath and you let the air out, then you can blow more air out. Okay, and that's going to be your expiratory. So we call inspiration, when you inspire, that's inhalation, so that's breathing in, and if you expire, that's breathing out. Okay, so those are the terms. So if you want people to expire, if you're in respiratory therapy, you also want them to inspire. Okay, fun, very inspirational. All right, so expiration. So the expiration reserve volume is what you can blow out after a normal exhalation, and then inspiratory reserve volume is what you inspire, or what you breathe in after a normal inhalation. Okay, so at the top of this tidal volume here, and at the bottom. So at the top, it's your expiratory or your inspiratory reserve volume that I just kind of covered up. But this is the inspiratory reserve, and this is the expiratory reserve. So they've got like 3,000 here and 1,100 here. It's different for males, females on average. But then you have, if you add all of these things together, this is the amount of air we have control over. Okay, so what do I mean by that? So we have expiratory volume, which is this area here. So we breathe as much air as we can out of our lungs. Then we have this normal breathing that we just do all the time, our tidal volume, and then we have this inspiratory. Okay, so if we add, and that's a lot. So if we add all of these together, that's as much air as we have available to us. So if we take a deep breath in, and then we blow all of the air out until we can't get any more out, that's what we call our vital capacity. Vital meaning living. So I think of it as that's what we use to live. Okay, so that's the air in our lungs that we have control over, and then we can live by controlling how much air we breathe in or out. So why do we point that out? Because there's air still left in our lungs even after we blow it all out. Okay, so there's still air in there. All the alveoli didn't collapse, we hope. So we call that the residual volume. Okay, so the residual is what's left after breathing out, after your expiratory reserve volume is blown out. Okay, all right. Let's see. What else? We're going to go over these again. There are like 3 slides dedicated to this. So your tidal volume, again, normal at-rest breathing. That's the up and down here, in and out, in and out. And then your inspiratory is at the top of the tidal volume, how much you can pull in. Okay, and then the expiratory reserve volume is what you can push out at the bottom, from the bottom of the tidal volume. Okay, and then you're back to the tidal volume. Okay, so those 3 things are kind of the big ones, and then you can kind of get other information from these others. So we have the residual volume. So we discussed that. That's how much is left in the lung afterwards. And I'll say that if you take vital capacity, and you add your residual volume, that's going to be your total lung capacity. Okay, so when we talk about lung capacity and increasing lung capacity, there are a lot of A lot of times you'll hear people say that, if you work out more, you increase your lung capacity. You don't really increase your lung capacity. Your lungs aren't really growing bigger. You can make better, efficient use of your lung capacity that you already have. You can expand your chest cavity and maybe make it more flexible so it can get larger. But really, your lung capacity doesn't change, just how well you can use the capacity that you already have. All right, so residual volume, vital capacity, we mentioned that. Functional reserve capacity. Now, functional reserve capacity is one of these weird ones that includes just these two things here. So it includes your expiratory reserve and your residual volume. So basically, if you just stop breathing and that's it, you're done breathing, that's what's left in your lungs. Okay, so so in order to get any of that, the functional reserve capacity, in order to get that out, you have to push it out. Okay, so you have to force that out. You have to squeeze down your chest cavity and kind of squeeze the lungs to push that out. Everything else is kind of something you're breathing air in, you're using your diaphragm, you're pulling it in, and you're letting the lungs just let it out. But to get that last, even to get that expiratory reserve volume, and then the residual just kind of stays there. But that's all positive pressure. It takes positive pressure to push that out. Okay. Let's see. And then we already mentioned total lung capacity, vital capacity, plus residual volume. So dead space. Dead space. Dead space is the volume of air in the airway and lungs that does not participate in gas exchange. Right? So it's just not, there's no alveoli. There are no respiratory areas for gas exchange to take place. It's just your trachea, it's your mouth, your nose, all of that area. So those conducting pathways, the alveoli, or I'm sorry, not the alveoli, but the bronchi, everything that's carrying it. So we call that the conducting pathways, just bringing air there. No respiration going on there. So we call it dead space. Now, that's anatomic dead space because there's really nothing you can do about that. Okay? Or that's just, a consequence of, being a breathing organism, is that you have to get the air down. However, there's a difference between anatomic dead space, which is just those conducting areas, and physiologic. Okay, so physiologic means that it maybe used to work and doesn't anymore, or it's in the lungs and it's not working right now. Okay, so what does that mean? That means that if there's some kind of lung damage that happens inside your lungs that won't allow respiration to take place, that's not a conducting pathway, but still there's no exchange. Okay, so the total volume of the lungs that does not participate in gas exchange. So if we have our lungs, and down here, let's say that there's some lung damage that's taken place right there. This is dead space. Space that isn't even readable, but that's dead space down here. So even though the lung normally would have the capacity for exchange, it doesn't in this case. And so we call that physiologic dead space because it's not conducting. It's not anatomic. It's usually like some kind of a disease. So some kind of a pathology, lack of perfusion. So you could have what's called a PE or pulmonary embolism that isn't letting blood flow to a certain area. So maybe it's clogged up with something and it's not feeding a certain area of the lung. So that's dead space. It's not helping anything. It's actually, just taking up resources and not really helping anything at all. Okay, so that's our dead space. So let's see, we're still in lung volumes and capacities, sort of, except now we're going to talk about breathing, ventilation. So ventilation and ventilation rates and what's going on with the whole moving of air. So minute ventilation, as you can maybe tell by the name, it's how many ventilations or how much volume, with the volume of air inhaled and exhaled in a minute. So we would compare this to like cardiac output. Remember when we talked about cardiac output, that was how much blood is pumped by the heart over a minute. Well, this is how much air is going in and out over a minute. So it's the total volume air inhaled and exhaled each minute. So that's going to be a pretty simple calculation. So if you breathe, so if your rate of breathing is you're breathing 12 breaths per minute, and each one is, this is normal tidal volume, is 500, then that's going to be 6 liters per minute. Okay, so that would be an average for a person if they're breathing 12 breaths a minute. Now, obviously, you can look at this and you can say, okay, well, if we're breathing 18 breaths a minute and they're all still 500, then it's also going, it's going to be more than that. So it's going to be more like, what is that, like 9? So that's going to be 9 liters per minute. Or if, you know, anyway, you can adjust it if your tidal volume is lower. So you can kind of see how that calculation is pretty, you know, it's not too bad. So, but that's minute ventilation, and then we have alveolar ventilation. So that's really, if we're talking about what really matters, then if you're talking about a patient that, you know, and you want to figure out how much air they're really getting, then you have to consider, remember we had that dead space. So if we have an area of the lung that isn't functioning, well, then we can't count that. Okay, because is it really, it's, I mean, they may be breathing in 500 milliliters, but there's really not respiration going on. So we call that alveolar ventilation. So that's minute ventilation, correcting for that physiologic dead space. Okay, so that's really all you do is you take the tidal volume. You say, oh, you breathed in 500 milliliters, but down here there's, you know, 50 milliliters of dead space. And so really it's only 450. Okay, and that's fair. That's fair. I mean, if you really want information about how much oxygenation is taking place and how much oxygen is available to the cells, then we can't count the areas that aren't being oxygenated. You can say, oh, he's breathing in 500, but if they're missing a lung, then, you know, that's, well, I guess that wouldn't really count because they wouldn't breathe in 500. But you get the point. If there's not actual perfusion taking place in part of the lung, then we can't really count that. All right, so when blood levels of carbon dioxide increase from increased metabolism, it is necessary for alveolar ventilation to increase so carbon dioxide does not rise. So what that means is that if you have a buildup of carbon dioxide, and we'll talk about that at some point, I don't think today, then your respiratory rate will have to increase. All right, so one more volume, forced expiratory volume, so FEV and FVC. So if you think about what happens and what matters, if you have a patient who is wheezing and can't breathe very well, you might see that if they try to push their air out, so let's say you breathe in, you know, 3,000 milliliters of air, and then you push it out. If your airway is constricted, it's going to take longer. It's going to be harder to push that air out. So we look at that over 1 second and we say, okay, so how much air, and you can do this experiment again on your own, how much air are you really pushing out in 1 second? Okay. Because that's going to tell me how wide open your airway is. So how long does it take to push, how much air can you get by blowing through a straw versus blowing through like a paper towel roll? You can, because the paper towel roll is a lot wider, you can push a lot more air in 1 second than you can through a straw. So that's what our FEV1 is, and it can kind of give us an indication. Is the airway obstructed? Okay. Is there some kind of something preventing that the air from moving through that airway? So forced vital capacity is just how much you're pushing out. So that can kind of go on a little bit longer. If you take a deep breath and you blow as much out as quickly as possible, then that's your forced vital capacity. All right. So, and the reason these are useful is they can indicate these pathologies. So if you have an obstructive disease, so that means that something is obstructing the flow of air out of your lungs. For some reason, you can't push air out of your lungs. Okay, it could be bronchitis, asthma, anything like that. And then you have restrictive diseases, and restrictive diseases mean that the lungs can't fully expand, which means, okay, your functional vital capacity is going to be affected. Or your forced vital capacity, sorry, is going to be impacted, because if you can't fill the lungs, then that's less air that you have to kind of mess with. So your vital capacity is going to be your inspiratory reserve volume. All that is going to be affected, and it's going to have an effect. And you can see on this graph right here that in just 3 seconds you've already breathed out all the air that you have. So your forced expiratory, your FEV1 in 1 second, that wasn't a problem. Getting the air out isn't a problem, but you've maxed it in that 1 second. You've almost gotten all of your air out, and there's just barely any more left. So if somebody does that and then they're done just, in 3 seconds, and that's it. Meanwhile, here you can see that, like an obstructive disease, so this is saying somebody with like COPD. COPD is a combination really of bronchitis and emphysema. And both of those, bronchitis really causes some obstruction of the airways. Emphysema can cause a lack of elasticity, but both of those things combined sort of prevent you being able to push air out. So in that first 1 second, you're trying to force air out, but remember, when you exhale, that's mostly elastic recoil. Okay? The diaphragm muscle is helping you breathe in, but it's elastic recoil, and then kind of, using some intercostal, which are the muscles between your ribs that kind of clutch down and kind of squeeze your lungs from the outside. But that's how you exhale. So when you're forcing air out, you have to depend on some of that elastic recoil. And if, like, emphysema breaks down elastin, it gets rid of that elasticity in your lungs, and so it's going to be harder to breathe it out quickly. But you can still see that after 3 seconds, there's probably still some air left in there. Okay, it hasn't quite leveled off like it has over here. Okay, all right. So let's kind of move move into the lungs. So we're going to talk about, we're going to start exchanging air and putting it in the lungs or in the body. So the alveoli, tiny hollow sacs whose open ends are continuous with the lumen of the airways. So most of the air facing surfaces of the wall are lined by a continuous layer, 1 cell layer thick, of flat epithelial cells termed type 1 alveolar cells. Okay, and we're going to see a picture of these on the next slide. I couldn't get all of them on one slide. And then, so this is what's actually making up the alveolus. Okay, so it's a cell. It's like those epithelial cells, but it's just a cell that makes up the alveolus. And there might be another one over here. Okay, that's the nucleus, and that's kind of what's making up these alveoli. And then there are type 2 alveolar cells that are also found in there, but these are kind of making surfactant. So they're their own cell, and they're making something called surfactant, which we'll talk about. Let's see what happened to my colors. Okay. So they're making surfactant that's going to kind of coat the inside of the alveolus. All right, so the total alveolar surface area is very large. Some say it's the size of a tennis court. Okay, that's the net. Or maybe, half a tennis court, which is also a lot, which basically means that you have so much surface area in your lungs that if you take one breath, a few liters of air, and spread it over an entire tennis court, thinly spread it over an entire tennis court, that's what happens with every breath that you take. Okay, so that's the surface area that are in these alveoli. Okay. So that's why you have such great gas exchange. And I'll tell you, that's also why that's a lot of surface area exposure for diseases to enter too, which is why we have why we have the cilia and the mucus, and then there are macrophages that kind of live inside your alveoli, constantly trying to prevent any kind of disease. So that's all going on in the background too. All right, so in some of the alveolar walls, pores permit the flow of air between, so we can kind of like what we saw with the lungs, with the heart, with sharing depolarizations, this is saying, okay, you know, I can share some of the air with everybody. So we kind of have air moving throughout, and then we have all of those capillaries trying to get oxygenated. So this is what we're talking about. So we have here, this is the type 1. So this is what I was trying to draw. So type 1 alveolar cell, and you can see that this is just making up. I guess it just goes to right there. So this is where one ends and another begins. And so this whole thing is just one cell around there. Okay, that's one alveolar cell. Then there's another one over here, and then maybe another one over here. I don't know. All right, but the point is that it's one cell layer thick, and then those are the type 1 alveolar cells that are doing it. Then we have the, where's the type 2? This is the type 2 alveolar cell, and that is what's kind of producing that surfactant that is going to coat the inside, and we'll see what the important things that surfactant does. But that's what's going to coat the inside with surfactant and prevent the alveolus from collapsing, okay? Because water is very sticky. If water gets in there, it'll collapse it. But water has to be in there for things to move around in. So the surfactant says, okay, water, chill. You're not going to collapse the alveolus. Not today. All right, and then we have a picture of the alveolar macrophage. So macrophages are in there because remember, there's no cilia in there to pull anything out. So anything that gets into an alveolus has to be dealt with. You have to kill it. And then you can see here, it's trying to show that here, the perfusion across these membranes. So here's a capillary right there. Oh, well, I guess this is a better capillary right there. And you can see that there's just this area of gas exchange that takes place. So oxygen is moving from the lung into the capillary, and carbon dioxide is moving out. So same thing over here, just kind of a blown up picture. So we have oxygen moving in there, and CO2 moving out. Okay, let's see. Basement membrane, very thin, nice access. Not much place to move across. I will tell you this though, that if you have inflammation that takes place, so if you have disease, then this area can widen. Okay? And if it widens, you have a more difficult time with gas exchange. Okay, so that is a real problem. So if you look at this, and it's something you'll talk about in pathophysiology, and you can kind of look at that distance and see that a wider area to cover means it's going to be more difficult to oxygenate your blood. All right, so pressure measurements. Respiratory pressures are always relative to atmospheric pressure. So atmospheric pressure is the pressure that's around us right now. We have air sitting on top of us and pushing on us all the time. We just kind of, so 760 milligrams, or we make it easy on ourselves and we say, okay, standard temperature conditions at sea level, it's 1 atmosphere. We just call it 1 atmosphere, and then if it's less than that, it's less, and if it's more than that, it's more. But we set that as our standard of 1. But that is equal to 760 millimeters of mercury. So we also use millimeters of mercury. Okay, so we can use both of these. Atmospheres just seems a lot easier, especially when we're comparing it to our atmospheric pressure. Now, depends on your altitude. Higher altitudes are going to have a little bit less atmospheric pressure in general because there's less air on top of you, and lower altitudes are going to be more. Okay, so if you're at a higher, like up in the mountains, it's going to be a lot less. All right, so what do we do with that? Boyle's law says that at constant temperature, you may have heard this in chemistry, at constant temperature, the pressure of a gas varies inversely with its volume. We probably know this kind of intuitively. So if we have an area that's this big and then we expand that area, keep everything else the same, but we expand it out, we're going to decrease that pressure. Okay, so that's less pressure. So the molecules are farther apart. And another way to look at it is if you have a balloon that is this large and you crush it, you're going to increase the pressure. So now you have all these air molecules that are super close together, and you're probably going to pop it. Okay, and it's going to explode. Okay, because you've increased the pressure. So that's what we mean. So it's proportional to the volume. What does this mean for the lungs? That means, remember, the lungs have no muscle tissue. So if the lung gets larger, this is dramatic, then its pressure is going to go down, which means that air is going to go in. As long as there's a place for air to go in, air will go in. So if we increase the size of our lungs, we've increased the volume, we've decreased the pressure inside the lungs, and air will passively move in. Okay. All right, and then we do the opposite. We squeeze the lung, pressure goes up, and air moves out. So that's why we talk about this. So a volume change leads to a pressure change. So higher pressure differences lead to greater flow. So if I want to breathe in quickly, then I pull harder with my diaphragm to pull to expand my lungs, and the air will come in passively. So the air comes in passively because of a volume change that we did actively. So what that means, the diaphragm pulled the lungs open, and air is just going to rush in because we've made the pressure lower in the lungs. All right, so greater flow of gases to equalize the pressure, so higher resistance leads to reduced flow. So if we put all of that together, and what we mean by higher resistance is maybe the width of the trachea, okay, or the bronchioles. So if we open our bronchioles, like in a fight or flight sympathetic activation, we open our bronchioles, then we have less resistance, which means that we have higher flow. If we close them, let's see, yeah, then we're going to have, flow is going to be restricted. Yeah, so there's resistance to flow based on how open they are. All right, so remember the gases always fill the container. So in a large container, the molecules in a given gas will be farther apart. We kind of talked about that here. Okay, if you have, whatever, 6, 7 gases there, and then you make it smaller, then you put the same number of gases. They're much closer together, kind of like moving from a big house to a small house. So, but that's going to cause, when they're farther apart, that's lower pressure. Closer together, that's higher pressure. So the diaphragm muscle pulls down on the lung. So we can maybe, in this terrible picture up here, we can say that here's our diaphragm muscle, okay? Diaphragm muscle, and when it contracts, it pulls down. Okay, and that's what's causing the lung to expand. This is kind of cheating because it doesn't make any sense, but that's what's causing the lung to expand, is that diaphragm pulling down on the space around the lung. Okay, so diaphragm muscle pulls down, increases the space around the lung, thereby expanding the lungs too, and increasing their volume and decreasing pressure. So again, I want to reiterate like 7 times that the lungs have no striated muscle. They don't do any of this on their own. They're just bags. Their expansion depends on the diaphragm muscle. So expansion is the only thing that we actively we have a muscle to pull the lungs open. We don't have any muscle pushing them closed. We can't, but we know we can blow air out, and that's because of contracting our chest area and making that area that the lungs are contained in smaller. And that's going to push them out. But for the most part, contraction or exhalation, expiration, that is a passive process using that elastic recoil of the lungs. Okay. And I'll say that 8 or 9 more times, I think. All right, pleura. So we've been talking about the lungs, and I keep saying, the area around the lungs. So that's what we're talking about with the pleura. So there's a kind of a sac, a double-layered serosa that covers the lungs. So it's kind of like if you take a balloon or something that's blown up and you push your fist through it, it kind of forms a double layer it would be a double layer balloon around your fist, okay, because you just sort of pushed it in. So that's what kind of happens with the lungs, is we have an outer layer out here and an inner layer in here. Anytime you have a double layer membrane, the one that's closest to the organ is also always called, just about always called, the visceral, and the one on the outer area is always called the, almost always called the parietal. Okay, so visceral versus parietal. Visceral is the inside, parietal is the outside of the pleura. So lots of things happen with this pleural. Very important. First of all, it's filled with fluid, and that fluid can build up. And this person has way too much fluid. It's not this big in comparison. So this is not to scale, because that's a huge pleura with a whole lot of fluid. This person has some like a pleural effusion or something going on, but it's good to look at and kind of make sense about how it's put together. All right, so the pleura produce fluid that remains in the pleural cavity. It lubricates the lung to prevent friction while breathing, so the lung can go up and down, or larger and smaller. And that pleural fluid, like with many things, can get infected. Okay, so a pleurisy can be infected or even damaged. So pleurisy is like when you have some kind of infection or damage of that pleural lining. Can result from pneumonia. Anyway, that can make the pleura rougher, okay, and can create friction. So maybe it's either so you've got granulation material in there, something. So it's causing friction, stabbing pain with each breath, and then there's that buildup of fluid, which can kind of hinder breathing. It can kind of push against it and cause makes it difficult for the patient to breathe, shortness of breath, that kind of thing. And then you order a chest X-ray and look and find it, and that's it. Then you try to get rid of that fluid with diuretics or whatever. I don't know. So let's see, intrapulmonary pressure. So we talked about. So what we have to do, what we're trying to do now with this slide, is kind of see how our diaphragm. So there's our diaphragm at rest. Okay. And then when it contracts, it straightens out. And what it does, as you can see, this is so much better than my pictures, which is why I have them. You can see that it made the lungs larger. Okay, so it pulled down, it increased the cavity, that space, and made the lungs larger. So you can see also in this picture, kind of dramatically, you can see that interpleural space here. See how large it is there? And you can see that the interpleural space became even larger, which means that you've created negative pressure. You're expanding that volume. You're creating negative pressure around the lungs. Okay, so this is our middleman. So the pleura is kind of the middleman of this. And so you pull on the pleura, and then the pleura pulls on the lungs. Okay, you create negative pressure in the interpleural space, and then that will pull the lungs even wider. The trick here is that the lungs are constantly pulling back. So the lungs, because they have all this elastic tissue, they're constantly trying to close. So if the pleura is pulling, and even when you're exhaling, when the pleura is pulling, it's pulling against the lungs. But even when you're expiring, when you're breathing out, it's still a negative pull, because your lungs, if left to their own devices, would just collapse. Okay, so we have these pressures that we call the alveolar pressure. We kind of mentioned that earlier. So that's our pressure in the lungs. Okay, so that's deep inside the lungs, in the alveoli, and how much pressure is in there versus atmospheric pressure. So if we have negative pressure inside the lungs, obviously air is going to move in the lungs because it goes from high pressure to low pressure. If we have higher pressure inside the lungs, it's going to push the opposite way. So it rises and falls with breathing and determines the direction. So when the pressure of the alveoli, so alveolar pressure inside the lungs, is higher than the atmospheric pressure, that means you're going to push the air out. And that's what we're showing here, is the relaxation of the diaphragm muscle and the elastic recoil of the lungs is going to allow that air to move out. Okay. All right, so airflow, the pressure gradient divided by resistance. We saw that earlier. So that's the difference in alveolar pressure and atmospheric pressure. So if we have higher pressure pushing out, then that's which way it's going to flow. But we can kind of figure that out intuitively. That's the plan anyway. All right, so pressure, what's going on in the pleura? So we already said that when your diaphragm contracts, it pulls the pleura kind of open, wider, it pulls on the pleura. But we also said that the lungs are kind of always trying to collapse. So what is the pressure in the pleura at any given time? Is it ever positive? Do we ever have, would it ever be positive compared to like the pressure in the lungs? It's always got to be negative. If it was positive, or if it was even the same as the pressure inside the lungs would collapse. Okay, because the lungs are always trying to collapse, and the pleura is the only thing preventing it. Okay, so if you put a hole in the pleura and say, okay, I'm going to open this up so air can go in there, then that lung's going to be like, nothing holding us back, and the lung is just going to collapse into a little tiny lung. Okay, so that's what happens. That's called a pneumothorax, and that can also happen kind of going the other way. If air from the lungs kind of moves into this area, then suddenly there's no pressure difference. There's nothing holding that lung open, and it will again collapse. So we need it to be a little bit negative all the time. So 4 or 5 millimeters of mercury less than the pressure in the lung, in the alveoli, in the lung. So that means the lungs are always being held open. It's possible because the lungs are elastic and constantly trying to collapse, but the pleura and the diaphragm prevent that. Okay. So if the intrapleural pressure and the alveolar pressure become the same, the lungs will immediately collapse because you need that negative pressure keeping them open. So this is a picture of that. So we can see let's see, interpleural space. This is very diagrammatic. So this is showing the lung. So alveoli is kind of equal to the lung. Okay. So, but we call it alveolar pressure, so that's why they drew it looking like an alveolus. Anyway, so that means that it is trying to, well, we'll just say it's trying to collapse all the time. Okay. And we only have, this is a little bit more negative out here, preventing that collapse, and that's just the normal way it functions. Then we have increase and decrease pressures, which is going to cause it to expand or contract. And this is showing that you've got about 5 less than inside your lung. So that's what we said, 4 or 5 millimeters of mercury less. So holding that lung open. And then we have pushed a scalpel through there, and we have said that we're not going to have negative pressure out there anymore, and the lung has collapsed. Okay. So because you're not holding that lung open, it just collapses. Okay, so let's see. So I have here expiration, inspiration, expiration. So this is all stuff we've kind of gone through. It's just kind of showing the same thing. Expansion, changing of pressure. If it's higher, then air moves out. If inside the lungs, if it's higher, the air is pushed out. And if it's negative pressure compared to the atmospheric pressure, air moves in. And that's basically what this is saying. So again, reminder, rest, expiration is a passive process. So that means the diaphragm muscle isn't involved. It's that elastic recoil of the lungs. The diaphragm muscle is just to pull it open. Okay. You can, of course, again, use those external intercostal muscles to kind of squeeze your ribs closer together and decrease that space. Okay, lung compliance. So compliance can be considered the inverse of stiffness. So if something is stiff, that means that you can't stretch it. Okay, so if you think of a bag or a balloon, if a balloon is very, very thick and stiff, it's going to be harder to blow it up. If it's very compliant, it can be very easy to blow up. Okay. So we kind of want to mix. We want good compliance, but we also want good elasticities. We want it to pull closed when it has to. So too much compliance can be not so good. Okay, so the greater lung compliance, the easier it is to expand the lungs at any given change in transpulmonary pressure. That's just the across the lung pressure across the lung. So there are two major determinants of lung compliance: the stretchability of the lung tissues, okay, and the surface tension. And we're going to talk about that in a minute. Surface tension at the air-water interfaces with the alveoli. So that's actually about 2/3 of your lung compliance is just that, the surface tension. So we have a picture here, and it shows green being normal. So that means that if we increase the pressure difference, so that means we are, so we're trying to breathe in, so we have negative pressure inside, then we are going to go from a minimum here all the way up to the maximum up here. Okay, so that's how much pressure change it takes to fill your lungs, right? To this volume. Okay, if we have really high compliance, it doesn't take that much pressure. So now we can see it only takes this much pressure, okay, to get your lungs to expand completely. Meanwhile, so that's increased compliance. So you would see something like that. Remember we talked about emphysema and how emphysema, there's the elastin proteins are broken down, so you don't have as much elasticity in the lungs. The lungs kind of start to be more like, I don't know, trash bags or something. And so they're very compliant. They open up, but there's not a lot of pressure in there to do it. So and you don't have as much exchange area, so you've got more dead space in the alveoli with someone with emphysema. So you've got increased compliance, but in that case, it's not a very good thing. And then we have decreased compliance. So that's going to be somebody with like a fibrotic disease, so scar tissue building up on the lungs. After a period of time with some lung diseases, you can get scar tissue, and then the lungs don't expand. It's harder to get them expand, so that's decreased compliant. They're not as compliant. Okay, so those are a couple of non-specific examples. All right, so water. So we were talking about surface tension. Well, water is very polar. So if you put water out on a table, okay, a bead of water, it will, and you just drop it there, it will form a bead. Okay, and we know this. We've seen this. We clean up water drops on our windshields and all this other stuff. But what happens is if you put a detergent in the water, it will flatten. It will flatten out, and the water won't stick to itself as much. Okay, and so that's what detergent or a surfactant does. Is it prevents water from sticking to itself as much. So water is very polar. It does that because it's got the, it's H2O, and this is, the oxygen is a little bit electronegative. The hydrogens are a little bit electropositive. So that means when there's another water molecule, they're going to line up like this. So they're going to be attracted to each other at these polar areas. So even though the molecule itself is not charged, it does have a partial negative and a partial positive side. So that's what causes it to stick together. So then what happens is if you give a surfactant, then a surfactant will it's got one side that's polar, and so it will kind of stick to the water molecule, and one side that's non-polar. Okay, so like a phospholipid kind of has that fatty acid chain on the end of it, and what that does is that it occupies the water molecules, so other water molecules aren't able to stick to them. Okay, that's what soap does. Okay, soap is kind of has that surfactant property, and it'll stick to the water, and then it'll stick to the dirt, and that way you're able to rinse dirt off your hands because then it goes out with the water. But the water isn't able to stick to itself is the issue. Okay, so surfactant can bind to water and prevent water molecules from sticking to each other, and that's what prevents alveoli from collapsing. So if you can imagine a drop of water gets in here, and I'll just make it blue just because it's water. So if a drop of water gets here, and if it's a small alveolus, it's just going to pull it closed. Okay, so all that water is just going to, all of this area is just going to pull that whole thing closed. However, if you put the surfactant on it, then the water will stick to the wall of the alveolus, but it can't stick to itself anymore. Okay. All right. So if it can't stick to itself, then it's not going to be as inclined to pull the alveolus closed. So we can see here what we were just trying to draw there. So remember, it's those type 2. So the type 2 alveolar cells are what's producing that surfactant. And that's going to prevent the water from pulling. Now, this is kind of, so we already said that 2/3 of the elastic recoil of lungs is caused by this surfactant water interface going on, all of this stuff going on. So what happens if you can imagine this area here in the alveolus getting larger, then these little molecules of surfactant are going to get farther apart, okay? Which means that you're going to have more water molecules. And now if they're really far apart, these water molecules are going to start pulling on each other, okay? Now, when it's smaller, then the surfactant are closer together, then the water molecules aren't able to pull on each other. But when your alveolus expands, you take a breath in, those surfactant molecules get so far apart that the water molecule starts pulling, and that's going to pull those alveolus shut. So, and then it's going to get smaller until it's back like this, and the water molecules aren't pulling on each other anymore. That's recoil. So that's the pulling closed of the lungs. So we have again another situation where water is constantly trying to pull your alveoli closed, but the surfactant is preventing it. But when we expand our lungs, the surfactant doesn't work as well, and the water will start pulling it closed. And your body uses that. It's like, okay, that's going to help us breathe out. Okay, so we let the water pull for a little while, then the surfactant gets close together, the water stops pulling, it's not fun anymore, and because the surfactant kind of ruined it. And then you breathe in, and it's like, oh, surfactants, all over the place, but water's able to contact each other again, and it pulls it closed. All right, so we have that two things going on. So the surfactant, we're saying it prevents the collapse of the alveoli. Okay, that's good. It's preventing the collapse, but it's also allowing it to pull closed, just not all the way closed. Okay, so that's what the surfactant does. It allows the water to kind of pull the alveolus shut to give us some recoil, elastic recoil, but it won't let it close all the way. Okay? So in infants, they haven't produced these type 2 cells, are not. They don't necessarily want it to be open because there's I shouldn't say in an infant, but before birth, in a fetus, I guess, that they haven't started producing that surfactant yet. So if they're born too early, then you have to get some positive you have to get some positive pressure or some artificial surfactant to put in there, so the premature newborn can breathe. All right, so gas exchange. So gases diffuse across thin alveolar epithelial cells and capillary endothelial cells to enter the plasma. Okay, so we're going to start talking about movement of oxygen through the lungs and down to the tissues. Okay, so we're going to kind of get into internal respiration. So steps of respiration: oxygen diffuses across. So just a big overview here. Ventilation, we breathe. Gas exchange, we have exchange of oxygen and carbon dioxide in the lungs. Okay, and then it moves through the system, left heart, after it's oxygenated, and then it goes to the cell. So we have gas exchange in the cells, which is cellular respiration, okay? And then it moves back to the lungs where it picks up more. So that's our gas transport. Now, one thing to keep in mind, so if you remember Fick's law, or if you don't remember, this is Fick's law. It is that in diffusion, particles move from a higher concentration to a lower concentration. So solutes move from a higher concentration to a lower concentration in fluid, or gases. Gases will also move from a higher concentration to a lower concentration, except they're gases, so we measure gases in pressure. And so, and since there are a whole bunch of gases, we have to think about each individual gas. So individual gas, so even though the concentration of oxygen is higher, if you have a high concentration of oxygen on one side, like in the lungs, and a low concentration of oxygen in the capillary, then oxygen will move from that higher concentration to a lower concentration. But remember, we also have carbon dioxide. Okay, so we can't just say it moves from a higher pressure to a lower pressure, because each of these contributes to the overall pressure. So we have to say the partial pressure of oxygen and the partial pressure of carbon dioxide. Okay, so the partial pressure of carbon dioxide is going to move, it's only going to move based on the concentrations of carbon dioxide, which is low in the lungs and high in the capillaries. So because the partial pressure of carbon dioxide is different in a different direction, it moves out of the capillaries into the lungs, and because the partial pressure of oxygen has a different quote concentration gradient, partial pressure gradient, it's going to move from higher pressure to lower pressure, but only for the oxygen. All right, so we'll just look at this. This is what I was just talking about, except that this is the partial pressure of oxygen. It's giving a number, and it's showing that the partial pressure of oxygen inside the capillary is 40. Partial pressure in the lung is 105, and so it's going to move into the lungs. And opposite for carbon dioxide, which is not as big a difference, but it still matters. But it's 46, and in the lungs it's only 40. So carbon dioxide is going to move out. And remember, this is external respiration. Okay, so that's what we're showing here. And then in the lungs, or in the tissues, it's the opposite. So we have internal respiration, and that's what's happening in the tissues, in the cells. Okay. So otherwise everything is what I just said. So how does it get there? How do things move around inside the blood? So is it just soluble and it just floats around in the blood? Kind of in some cases, but not really. So if we look at the solubility of oxygen versus carbon dioxide, we can see that here's oxygen in air, and this is oxygen in water. Okay, so we can see that oxygen is kind of soluble in water, so some of it might be there, but we can see that carbon dioxide is a lot more soluble in water, which gives some of our differences. But so we have to think about, well, if oxygen isn't very soluble in water, and that's how we're kind of blood is made from water, then how are we getting oxygen moving around? And that's because it is primarily carried on hemoglobin, a molecule called hemoglobin, which we'll look at. Carbon dioxide, yeah, it's soluble, but the trouble with carbon dioxide, or a feature of carbon dioxide, is that carbon dioxide will react with water, and it forms, let's see, H2CO3. All right, it forms a molecule called carbonic acid, which then can break down to, it loses a hydrogen, and it becomes HCO3 minus. Remember this from chemistry, and that is bicarbonate. Okay, so that's the issue, is that carbon dioxide, even though it's soluble, will react with water, forms that carbonic acid, and then it's actually this is actually an enzyme that causes it to react, and then it's stored there as bicarbonate. All right, and then nitrogen, most, like 78% of the air we breathe is nitrogen, and but that's not very soluble at all. although it can build up and it can cause like crepitus and your knuckles to pop, and it can cause the bends if you change your, like, come up when you're scuba diving. If you come up too fast, the nitrogen will expand and can cause bad things to happen. All right, so internal respiration. So oxygen diffuses from systemic capillary blood into the tissues, and so they're constantly, cells are constantly using oxygen to make ATP in the mitochondria. Remember, the mitochondria, powerhouse of the cell, is making ATP. And then you have glycolysis out here, and if you don't have enough, so this is what oxygen is for, is to kind of make ATP in the mitochondria. If you don't have oxygen, okay, no oxygen, then you end up producing something called lactic acid, and that's less efficient because you only make a couple of ATP with glycolysis in all those steps. So even though it can do it without oxygen, it's not very efficient. Okay, this is better. Okay. So that's also how we can tell if tissues are being starved of oxygen, is because we have high lactic acid levels. So remember that. Okay, so signs of infection, sepsis, any kind of like a heart attack, you know, is where muscle cells will die, or anywhere there's hypoxia, lack of oxygen, you'll see increased lactic acid levels. Okay. All right, and it's all because of mitochondria, which are the powerhouse of the cells. All right. So carbon dioxide, so same thing, diffuses from tissue cells into systemic capillaries, and cells are constantly making that carbon dioxide. So, you know, again with the mitochondria, as it's making this, it's also making carbon dioxide and glycolysis too. And it's all making it from glucose. Remember your 6-sided glucose molecule? That's 5. Okay. All right. Okay. So at rest, only about 25% of the available oxygen is used. We're going to see that in some graphs that we're going to look at. Deoxygenated blood would retain 75% of its oxygen capacity. But that's going to make the differential less when it's trying to drop it off, and so, you know, it's going to make it less inclined to leave and move into the cell. So let's talk about hemoglobin. So 98% of oxygen is carried on hemoglobin, and so we've got this great system really designed to carry oxygen. Okay, so that's what the whole purpose of hemoglobin is. And it's kind of complex. So there are 4 subunits. It's a large protein made from 4 subunits. And when we talk about proteins and subunits, so subunits are like, it's a different gene that translates. Okay, so translation transcribes the protein. So, and then those individual proteins come together to make a larger protein. So that's what we're talking about. So we have smaller subunits, which are here. You've got the beta subunits and then the alpha subunits. We call them alpha chains and beta chains, but they all come together. And in adults, you have, like I just said, the alpha and beta chains. In fetal, they have gamma chains instead. Okay, so instead of beta chains in a fetus, you're going to have gamma chains. All right. Now, that's going to be relevant. But let's first look at how the hemoglobin is put together, because that's also relevant. So you have the heme groups, okay, or not, I'm sorry, not the heme groups. You have the hemoglobin subunits, and in the middle of each of these subunits, we have a heme molecule. So that's just a molecule. So we have the heme molecule that is sitting there being held by the hemoglobin subunits. And then in the middle of the heme molecule, we have iron. Okay? And very nicely, the oxygen sticks to the iron, and that's how we carry oxygen around in our blood. And these red blood cells are packed with hemoglobin. They've given up their nucleus. They've just shortened their life just to carry as much hemoglobin as possible. And each hemoglobin molecule, yes, if you did the math, can carry, oops, and I'm not doing the math right, can carry 4 molecules of oxygen. Okay? Because there are 4 subunits with 4 hemes, and at the center of 4 hemes are iron, and each iron can carry 1 oxygen. Okay? All right, so iron is very important. That's why a lot of people, if they don't make enough iron, then when the blood cell, when the bone marrow is trying to make red blood cells, that if it doesn't have enough iron, it just kind of makes a bunch, and they become kind of big and bulky and break easily, and that's just not good if you don't have enough iron, because it's waiting for the iron, and they kind of become what they call macrocytic. They become large, and they can break. Okay, and then you become anemic. That's what anemia is. It's not enough red blood cells. All right, so let's look at how this works. So here's our red blood cell. We have oxygen, binds with the hemoglobin. Okay. So, let's start at the beginning. So this is moving from the alveolus. So it moves here. And remember, oxygen is not very soluble in water, but it's a little bit soluble in water. So what happens is that the oxygen initially moves into the blood and then is very quickly swept up by the red blood cells, and it's stuck to hemoglobin. Okay, so about 98% of the blood, of the oxygen is in hemoglobin, and 2% is dissolved. All right, so that's what happens near the artery, or the capillaries in the lungs, and then in the tissues, the capillaries in the tissues, well, what happens is the oxygen begins to fall off of the hemoglobin and will move down its concentration gradient, because there's less oxygen near the tissues because they're using all of the oxygen. And so you have a situation with where the oxygen kind of moves off of it. Okay, so let's look at what this looks like on a chart. Okay, so we can see that. Well, we're kind of looking at it. We're talking about the saturation. So as we have higher concentrations of oxygen, we have more and more of it being bound to hemoglobin. Okay, so that's what this graph is showing, is that higher partial pressures of oxygen, so like you may see in the lungs, okay, or in the alveolus, we'll see higher concentration. We'll see more on the hemoglobin, so more attached to the hemoglobin. And then where you are in the tissues, then the hemoglobin or the oxygen will drop, and you have less on the hemoglobin because it moves off and it moves into the cell tissues. So this is what we call the oxygen hemoglobin dissociation curve. So this is the same thing, and it's saying at a resting cell you'll have about 40 millimeters of mercury of oxygen on the in the blood and in your hemoglobin. Okay, and then where you are in the alveoli, it will be completely saturated. Okay, almost completely saturated. So you can see when we talk about how it maintains 75%, it's only 75% bound. That's what we're talking about. We're not looking at the partial pressures by number. We're saying how saturated is this hemoglobin with oxygen. And you can see that it's almost 100% saturated starting out as it's moving out of the heart, left side of the heart. But then as it leaves the tissues, it's only 75% saturated. So why does it leave at all? Okay, so we said that it kind of goes down a concentration gradient. So why, when it gets to the cell, does oxygen fall off of the hemoglobin? It seemed perfectly happy there, but it really wasn't. It was fine, but things change, you know. And so as the oxygenated hemoglobin gets to the cell, things are going on in the cells. Okay, so first of all, one of those things, that's a cell. One of the things that goes on in the cell is it's producing a lot of carbon dioxide. Okay, so lots of carbon dioxide in there. And what did we say happens to carbon dioxide when it's dissolved in water? It reacts with water, okay, and it forms something called, I always have to count my molecules when I do this, carbonic acid. Okay? And then, yeah. And that carbonic acid is converted to hydrogen and bicarbonate, O3 minus. This is the issue right here. This hydrogen that's being produced, so more carbon dioxide pushes this equation in this direction, and we have a buildup of hydrogen, which means our pH, our fluid is becoming more acidic, which means that we are in a low pH situation. Now, what that means for the hemoglobin saturation is that at lower pHs, where it's acidic, it takes more oxygen to saturate it. Okay, so it loses affinity for oxygen. So oxygen is not bound as tightly where it's acidic. So you can see here that, you know, pressure changes are going to make it just run all the way up. It's saturated already here at, you know, at whatever that is, 70 whatever, 70. But it's 70 millimeters of mercury at a low pH. It's only like 85% saturated. So it loses its affinity. And that's convenient because that's the same place where we want oxygen to leave, okay, and move into the cells. Okay, so the oxygen binding affinity is lower at lower pHs where it's acidic, which coincides with the environment of the cell being more acidic because of the metabolism that's taking place. That mitochondria, which is the powerhouse of the cell, moving oxygen, taking oxygen in and producing carbon dioxide. Okay. All right, so environment near metabolically active cells tends to be more acidic. pH saturation curve favors dissociation of oxygen from hemoglobin near those metabolically active cells. And then oxygen falls off, moves down its concentration gradient and into the cell. So how does this happen? Hydrogen binds to histidine residues on the hemoglobin subunits. So remember those hemoglobin subunits? They have these little histidine. They've got a lot of amino acids, but one of those amino acids is histidine, and hydrogen will stick to those. Okay, stick to those, and then the oxygen that's being carried in there falls off. Okay, so lowers affinity. So there's some other things that we're going to kind of look at here that also change affinity. Temperature. So remember, when it moves to the right, then the temperature, then that means lower affinity. So it doesn't bind as tightly. So at higher temperature, oxygen also, the oxygen hemoglobin connection is also lower. Let's see, maternal and fetal hemoglobin. So again, if we compare maternal hemoglobin, we can say it has lower affinity, so it binds oxygen less tightly. And fetal hemoglobin has higher, so it wants the oxygen more. And that's convenient because, you know, where mom and baby, you know, they don't share blood, but it's close together, then baby will, or fetus, will move the oxygen and it'll bind that oxygen more tightly than mom. Okay, so baby or fetus gets more oxygen. Okay, so how does that work? Well, there's something called DPG, diphosphoglycerate, that is produced in the red blood cells, and it just kind of says, okay, hemoglobin, you're lacking oxygen, so I'm going to make that more comfortable for you. Okay, so it reduces that binding affinity of oxygen. It stabilizes deoxygenated hemoglobin. Says, okay, you know, you can live without oxygen. That's fine. Well, this doesn't happen as much in the fetus. So it's not as stable, it's not fine, it wants oxygen, so it has higher affinity. Okay, so we can see that in the graph up here with no DPG, you have low affinity, or I'm sorry, you have high affinity, and with the DPG, you have lower. Remember, lower affinity is to the right. All right, so here, that's kind of what's happening. So this is our alveolus, external respiration. Oxygen moves in, is bound to the hemoglobin, and then at the cell, certain changes, particularly acidity, pH. It loses its affinity and moves down its concentration gradient to move into the cell, or partial pressure. All right, so let's see. So these are the kind of drawing out the things that I've been talking about. So this carbonic acid bicarbonate, the carbonic acid bicarbonate buffer. So this is your blood buffer, basically. So more acid, more hydrogen is going to push the equation that way. Less, more. Let's see. Yeah. So so here, let's just look. So carbon dioxide, we'll start from left to right. So carbon dioxide, this is the equation we've already been talking about, is going to push the equation that way when you have more of that, which is going to be a buildup of hydrogen. Okay. Now, what can happen is, and so that's going to be low pH. But we also know that bicarbonate is a very, it can act as a base. So if you start adding bicarbonate, then what that will do is it will occupy these hydrogens and it will move the equation to the left and your pH will go up. Okay, so adding bicarbonate will cause your pH to go up. Adding carbon dioxide will produce more hydrogen, which is going to cause your pH to go down. Or you could just add hydrogen, and that would also cause your pH to go down. All right. So increase in carbon dioxide from metabolic processes increases bicarbonate ion, which is how carbon dioxide is stored in the blood. Okay. So that is the primary way, and we'll see that in the next slide. And you need to know how much. So increases hydrogen, lower pH. Yeah. And it says here, can you guess the ventilation response of a patient with diabetic ketoacidosis? Your lungs sort of know that they have this power with carbon dioxide. Okay, they don't know, they don't have a brain, but something knows, because if your carbon dioxide levels increase, your rate of breathing will increase. So you can get rid of, so you can blow off that carbon dioxide. So what that means is as your pH becomes lower, your respiratory rate will become higher, okay? Because you're trying to blow off, you're trying to push this equation to the left. You're trying to get rid of carbon dioxide. If you get rid of carbon dioxide, it just keeps moving to the left, okay? Now, if you hold your breath, then your buildup of carbon dioxide and your pH will go down. Okay, so at a low pH, you want to increase the respiratory rate. So I put this here, diabetic ketoacidosis. We don't necessarily need to know what that is, but it is where your pH goes down and your blood becomes acidic. Okay, and the response of that is your respiration rate is going to increase because you're going to push it the other direction. Okay, now if you vomit a lot and hydrogen is leaving your body, then it's going to push it the other way. Okay, so that kind of stuff happens. So, but right now we're kind of focusing on carbon dioxide transport in the blood, and we see here that 70% of it is carried in that bicarbonate because of this equation right here. CO2 plus H2O, and then we have the bicarbonate, and that's how it's being held. Okay. And also of note, we can see that the bicarbonate doesn't build up in the red blood cell because it's moved out by an exchange by chloride and bicarbonate exchanger. But we're not too concerned about that right now. But in case you're curious of why bicarbonate is constantly being produced, but I mean, how does it get out of the cell? That's how. So 23% is carried on hemoglobin. So yes, hemoglobin has some affinity for carbon dioxide as well as oxygen, and 7% is carried dissolved in the plasma. Okay. All right, and then it goes in and out from the carbon dioxide from the tissues, and then it's moved out to the lungs. All right, and that is it.